So, okay, our recording starting. So thank you everybody to this new installation of the International Legume Society webinar. I'm very happy to be here today with Diego Rubiales from CSIC. His story is very intertwined with the International Legume Society. And I'm very happy today we are going to talk about disease resistance in legumes in general, and we're going to use Pisum as a model species. As always, this webinar is recorded and you can find it online. And as we usually do, you can ask your question in the chat and I will read them at the end for Diego. So without further ado, please, Diego.
So good afternoon, everybody. Well, good afternoon from here, from Spain. If there is some good morning, if there is someone in the audience from America, or good evening if we happen to have someone from Asia or Australia. I am here to speak today about legume breeding for disease resistance. We will focus on pea. We also work a little bit on fava bean, on lentil and other legumes, but we will focus on pea. And even in pea, there are many different diseases. We will focus on the use of wild relatives. That is what we used to do when there is no resistance in the crop itself. Breeders are probably there are breeders in the audience that know better than me that there are many different breeding priorities, many targets that the breeder have to satisfy. The consumer has to satisfy the processor, the society, and the farmer, in which there are many aspects: lodging, herbicide tolerant, yield. But just to complicate the thing, we are focusing on pests and diseases that are important, but alone is not sufficient as to have a good variety, because it should be resistant, but also having good quality, good size, good different properties. Even the diseases, which disease to focus? Well, there are many different, just thinking, for instance, in peas, many different pests on diseases, host relatives, prevalent and important, depend on the agroecological condition and even in the farming system. So, for instance, in the Mediterranean region in pea, the major constraint is broomrape. That is a parasitic weed that probably people from other areas has never seen. On the contrary, in other areas is aphanomyces that we don't have in the Mediterranean condition because the soil is too dry, or many different aspects. So we should, you know better than me, that a breeding program takes long, 7 years to produce a new variety, a few years more to register it. So you have to be prepared for what is coming. And this is the issue. As I said, I work at Córdoba in the very south. It's typically Mediterranean region with very mild winter, rainy, and extremely dry and warm summer. So our crop, we plant, let's say, the peas in December and harvest by May. If you see here, this is the picture of what I see from my window. This is a picture of this morning. I have also in the background, but for us, this is very cool day. So the constraints that we have are not the same that people have in Germany, in Canada, or in other areas. So we have to address our priorities, but we are also covering also other diseases or pests that are from other areas, because we need to breed for other areas, and also because we need to be prepared for what might come with the global climatic change. That is something that we are experiencing. Just allow me that I presented the area just to show where I work. I work at Córdoba. That is an rather small city with 300,000 inhabitants and with a small university, but strong in agriculture. And also Cordoba has a long history and is very nice touristic place that was important during Roman Empire, was the capital of the Roman Betica, but Cordoba was particularly important several centuries later. So by the 10th century, Cordoba was the largest and wealthiest city in Europe, and almost all the Iberian Peninsula was under the Caliphate of Cordoba. That was only then. Now Cordoba is just a small city, as I said, with a small university, strong in agriculture, strong in tourism, but little else. But Cordoba is the city with more UNESCO heritage sites in the world. I invite you to visit Cordoba, not in the summer. So coming back to the point of our presentation today is how to breed for pests and diseases. And for me, breeding is very easy when you have good sources of resistance, you have good, affordable, sufficiently accurate screening methods, and you have the breeding tools in hand. So three things that we seldom have. But if we had, it will be very easy. We just need it, we'll need time. And particularly the problem is when we have problem, a pest or a disease for which there is no known source of resistance. And then is when we have to go to genetic resources, and if even there is not present, then we have to go to wild relatives. That is what we experienced it in beef, and it's why we started. Fortunately, we have very large collection of genetic resources in different gene banks. So different sources say that we have circa 300,000 P accessions in different gene banks. Probably many of them will be duplications, and about 1% of them are wild relatives. So this is impressive resource, but the problem is that in most of the cases that they are just stored and they are not properly characterized for the traits of interest to us. Probably there are some descriptors, but we need to do the screenings for our traits. Unfortunately, as I sit here, in the secondary gene pool, so in the wild relatives, sufficient variation, sources of resistance has been identified for a quite number of stresses that is summarized here. I will go now in more detail, one by one. So the good news is that we have impressive collection of genetic resources of wild relatives, and that resistance to most stresses have already been identified. Some levels of resistance, not always complete resistance. But then the challenge is how to use this, to introduce this in a breeding program. Something that I will show examples in which this has been successful. The problem is that let's say, it's one of the most important legumes, but still is not as important as crop as rice, wheat, maize. So this pre-breeding, introducing resistances from wild relatives is often hardly affordable by the commercial breeding programs. So here the interaction of the public sector is most convenient. I presented about the genetic resources and now the tools. The genetics was born with, modern genetics was born with P, the laws of Mendel. But then during quite some decades, P as other legumes were lagging behind other crops. But in the past decade, this has progressed impressively. So now, only if you remember, the first P reference genome was released only 2019, so 7 years ago. But then, since then, there have 2 more genomes have been released, and now there are several pangenomes in progress. With so all the genomic resources, tilling population has been progressing very rapidly. Fortunately, we are facing very similar situation with most legumes, and this is thing I will not go in detail in the next 3 slides, but it's just to introduce. So marker-assisted selection is already achieved, is already done for monogenic traits in pea for different diseases like Fusarium wilt, powdery mildew, PNH mosaic viruses, where there are single genes markers. So they have been used for marker-assisted selection. Even for more quantitative traits, marker-assisted crossing is being attempted by several groups, and genome-wide association studies is nowadays a routine for most in pea for most diseases, quality traits, and other stresses. And even we are the level of doing genomic selection for more complex traits. So there are already studies on yield, drought, ascota, and RAS. So this is progressing very rapidly. But now let's focus on the use of wild relatives. But as I said, at times it's complicating everything a lot because you cross with something that is of very poor agronomic quality and you introduce many different undesirable traits. But in many occasions it's needed because you don't have the trait, the resistance in the crop or in the all cultivars, or it's always better to cross a good cultivar by another cultivar, so you get something better. But here we have to cross a good cultivar with something that is very, very, very bad agronomically, but has got the trait of interest. And this is the situation that, sorry to start again, to talk again about broomrape. I will mention a number of pests and diseases. I cannot afford more than 1 or 2 slides of them because there are many in which we have been working. As I mentioned, broomrape Orobanche crenata is a typically Mediterranean problem, and is the reason why I started to work on legumes. I had already in 1995-3 a permanent position to work on diseases on wheat and barley, but then I saw a problem of pea and with broomrape, in which nobody was working to my knowledge. So I started little by little working on that, and now during quite some years was my main activity. And here is the example where we started screening all the commercial cultivars or larger plant of Pisum sativum, and we didn't find any resistance, so we had to go to wild relatives. So I cannot focus here, but here I will present more than 70 scientific publications on this aspect, broomrape, pea, or other legumes, identifying sources of resistance or validating or adjusting breeder-friendly screening protocols. So we even try to then understand, characterize the resistance identified at the genomic and proteomic tools available at the time, and to identify markers associated with the resistance. So first with biparental populations, in which I always say that we always complain that we don't have enough genetic resources, genomic resources, and we need better markers, what is true. But I always insist that probably we even need more improvement on the phenotyping accuracy. And by this, I don't mean high throughput output phenotyping, very expensive phenotyping platform and tools, but something affordable. So here in this paper, we saw identify a number of QTS when screening the broomrape emerged in the field and comparing with the following the different mechanisms of the resistance in vitro screenings and identified some in common and other not. But still, these identified QTS still, let's say, small, what we call a small, explaining insufficient low number of the phenotypic variation, not justifying a market-assisted selection. More recently, we have done a genome-wide association study in a collection mainly involving wild relatives that has been completed. It is not visible clearly on purpose because this paper is still under evaluation, is submitting. So it's just to say that this has been already completed, but I cannot give much details. So all this, I summarize a long process of studies on broomrape in which we have progressed a lot in identifying and understanding the resistance, but we are not yet at the level of being able to use marker-assisted selection or to do it in preview. Fortunately, they had let's say, a kind of duplicate perspective. So besides doing science, I was doing breeding and selection at the classical style. So identifying sources of resistance, making crosses, and selecting every year under very heavy and uniform infestation. And this was successful. So in a few years, we had a first generation of very, as you can see here, very resistant lines compared to the surrounding checks. So we, without understanding the genetic basis on the marker, we were able to introduce the resistance into P background, but still, and these lines were of moderate yield. So after this, we dedicated a number of years just saying, okay, we have the resistance, but this cannot be cultivated. So now we have to improve the standing ability, the yield, and so during several years more now crossing with among elite lines, we have now a set of cultivars, as you can see here, that compared to the checks that are resistant and very productive and adapted to our conditions. So with this, more or less, we decided not to stop with broomrape because, well, still we have many scientific questions still unresolved, but the solution to the problem is here. And we decided to concentrate on other diseases because, okay, we have a cultivar that is resistant to broomrape, that is yielding well in our conditions, but probably is not good in other areas where broomrape is not the problem. So we need to introduce also resistance to other diseases. Only recently, when we had decided not to concentrate more on broomrape, we have been engaged in IP project in which our focus, our commitment is to come back to the work on broomrape in P, and we are coming back very, very happy. So as I said, this was broomrape. Another example of very successful introduction of resistance from wild varieties was powdery mildew, Pisci, and then we did work also a little bit on trifoli, that is also affecting P, where we started by screening large collection and identified a number of accessions that were very resistant, characterized the resistance at different levels, even histology. But the good thing is that we identified in P. fulvum a new gene, ER3, that is a dominant gene that, by crossing selection, was introduced into elite cultivars. As you can see here, it's very phenotyping for powdery mildew in the field is very easy. You can see even from the window, but then still, it will be good to have markers, because if you want to combine that resistance with the ER1 and ER2, that are the only other two genes now in P. So there was involved identification of markers, and GWAS in the panel of before, and RNA-seq work in progress that I am not presenting is from Manuel Alejandro Jiménez Vaquero. You will learn very soon of his progress because he's extremely good student. Our priority, I mean, scientifically, what I like most is RAS. I did my PhD in RAS in barley and wheat. So we work a lot on RAS also in lentil, in fava bean, but RAS in pea is not so much a problem, but we did a lot. Here is not as just as before, as it is not just a problem, so we just did more scientific work identifying sources of resistance, characterizing the histology, even different screening methods, even developing tools for with images to facilitate the screening. There were QTL mapping using a real population of peas within Pisum fulvum, so 2 Pisum fulvum accession, resistant and susceptible, and this was done first with rapid marker and later on with DARSEC. And more recently, a GWAS study identifying SNPs and that associated with the resistance. But as we said, here as there was not the demand of the breeders on this disease, we did not focus too much on the introduction of resistance, but we can say that earlier we had only slow, moderate levels, like you can see in this picture of slow rusting, so non-hypersensitive resistance, and only after extensive search of many accessions, we have now also hypersensitive resistance that might study in more detail in future. Another disease that in which we paid a lot of attention because it's very severe in other areas is ascocaite blight. The name has changed. We called before Mycosphorella, Pinode, then Dimela, now Peronella. Well, the point is that we are starting again as in P, screening a large collection where most or less in P from 1 to 5, most of in P, all that we have is 4 to 5. Even the more resistant P cultivars had only a 4, and we identified much better resistances in the wild relatives, and particularly in some of them. This resistance we characterize by different genomic and proteomic tools that are adapting to what was available at the time. First microarrays, then deep superset, then maize, also proteomics, and we use a real between Pisum syriacum and Pisum sativum and identify a number of QTS and compare them with the QTS identified by other teams in P. Also, there was another work on another ring with Pisum fulvum by JAB. So this was compared, but at the end, always frustration is that contrary to Poderemyrdio and to broomrape, in which we have seen a clear progress, introduction of the resistance for ascocaita blight, we and forgive me if I say something wrong, but to my understanding, nobody else in the world has progressed very significantly. So we are still only having moderate levels of resistance, and we have been unable to introduce this resistance, this strong resistance from these wild relatives, using the, let's say, the genomic-driven approach. So we are coming back to the old-fashioned that we didn't use in the past. So crossing and selection and the very heavy infestation level, and we are starting to see some progress for us. And also there is the GWAS study and we are repeating now. So the work has been done in the 5th. We are repeating another season and under growth chamber conditions. So this is the PSD of Manuel Alejandro. It's powder mildew and ascochyta blight. Fusarium wilt is a disease in which there are races. There are genes identified for resistance to race 1, not that much for race 2. There were only quantitative resistance. So we have done 2 PhDs on this. In a first one, we're identifying sources on resistance and characterizing it even at the level of histopathology and proteomics. So this is the work of Mustafa Bani from Algeria that now is back in his country, co-supervised with Nicolas Rispel. And a more recent study was the GWAS that I mentioned before, was the PhD of Wohor, that is here, and who did the GWAS for broomrape, that, as I said, is completed, but just submitted, and also for Fusarium oxysporum, for races 1 and 2, that I said is not clear the picture because this work just submitted, not yet accepted. So hopefully you will see the full paper published very soon. And now we come to pests in which we knew, and we still know very little, but where is becoming more and more bigger problem, particularly with the tendency to reduce less pesticides. So in practice, here in my region, farmers, we never had to apply for this fungus, but for aphid, if you don't apply with pesticide, then the crop is destroyed. We need to work on that. So, as I said, I don't want to be the best specialist in a topic. We want to do something helpful for the pea community. So, as I said, for me, if I had plenty of money and no pressure, I would work just with rust. But rust is not a priority for anyone. So if people come to me, farming, saying that aphid is a problem, so we have to do something. So there was a first PhD by Thais Fernandez, now is tomato breeder in the Netherlands. So we identified and published different sources of resistance to aphid based on field screening, very complex, and you had to do many replications, but also choice and not choice tests under controlled conditions. With this, we were able to identify QTLs for resistance in using the real population involving full bloom, and there is a GWAS genome-wide association mapping study in progress, in which we have already data from 3 field seasons, and is complemented also with the screenings under control conditions. Hopefully, very soon you will see better results. And this was the aphid in only one slide, and then we have to one even more difficult, that is P. weevil, Bruchus pisorum, that is very easy to screen under natural infestation the percentage of seed infestation, but to manage the insect and to inoculate, let's say, it's very difficult. Even with aphid, we can culture and population on living plants and in a growth chamber and then release them. For the Bruchus is far more complicated. So in the PhD, as I mentioned before, of Thais Fernandez was aphid and weevil. So there was sources of resistance identified based on the percentage of seed infestation, but also on the stage of development, cutting seeds one by one and scoring how developed was the larvae inside the seeds. Also, QTLs identified by us using our repopulation, and this was also seen in Australia and also in Pisum fulvum. And there is more recently, we completed the GWAS study, identifying a number of markers associated with the resistance. As I said, we need to. Each time I go to a meeting, I say, well, we have identified resistant to broomrape, resistant to powdery mildew, and people say, see, oh, yes, very interesting, but what about Aphanomyces? I say, well, Aphanomyces, we don't suffer from Aphanomyces, but okay, Aphanomyces seems to be the major concern in the larger pea producing areas like Canada, France, because there is no good resistance. So, well, we have started to work on this. So there is a PhD underway with already a publication listing sources of resistance in different wild relatives and comparing different screening methods, the standard in the roots, screening the root, with comparing with foliar screening of the foliar symptoms, and there is work in progress in characterizing the more resistant affection with measuring different photosynthesis and different parameters, and screening the GWAS panels. So hopefully very soon we will have nice results. And nematode. Nematode, we know very little, but we are very fortunate that we have a very good nematologist team in our institute. So we were lucky enough to engage the Juan Emilio Palomares to co-supervise a thesis that is undergoing and is now developing the screening methods to screen for, that is very complicated, for Platylenchus tornea mediterraneus and even different work looking for other nematodes, and this is the genetic studies is starting now. And only this. With this, just I want to thank you for attending and the different projects in which we are involved now and are supporting these studies. Thank you very much.
Okay,
Should
thanks.
I stop presenting?
As you prefer. Okay, thanks, Diego. It was a pretty impressive survey of your experience and your results on different kinds of pathologies and parasites. As usual, we take questions at the end of the presentation. Just before that, I remember that I put in the chat a link with a form where you can do actual feedback for us, the organizer of the forum, of the webinars. So are there any questions? Okay, I think I will break the ice just to see where it's going. So you started saying that there are currently a few pangenomes going on in pea. What do you expect to be the improvement, the contribution, and the value of having pangenomes compared to traditional genomes, or regarding resistance, but in general for our breeding efforts?
Well, this is not my specialty. I am not a genomic person, but I am very happy that this is progressing, particularly because, well, as I said, the first reference genome was released only, I think, 7-8 years ago, and then a second, then a third, and then I'm aware of at least a Canadian consortium in which they are doing the pangenome. And the good thing is that they are also, what is interesting to me is that they are including some of the resistant accessions that I identified. So they are including, so we will have, when doing our genetic analysis, we will have our accessions already with the genome. So I am eager to see the results. So this is also, well, as I said, I am aware of a Canadian and also in the cruising crop-wide relative European project, which we are involved. There are different species. P, they are from one of the partners is involved in this and doing they commit at least to do three genomes of the wild relatives of P. I think Paolo also.
Thank you. Let's raise them from Paolo and then a couple from the chat.
Thanks. Hi, Diego. Thank you indeed for the very interesting seminar. The pea weevil, the Bruchus, is certainly a very, very big problem in pea and also other legumes. I wanted to ask you, is the, let's say, more tolerant material, is this tolerance related to some particular characteristic of, like, for example, very high content of tannins, something that in a way that it may also have a negative impact on the seed quality, at least for certain users?
Yeah. We tried to study the correlation because this was screening it under field conditions during 4 seasons. So it's the seeds collected from 4 seasons, and we had all the agronomic trait as well of those affections, and tried to run also a correlation. So there was a small correlation with, as you said, with dark column, that is more, or the seeds that is associated with tannins or whatever, but was not very strong. So it was easy to find more resistant affection with white seeds from white flowers. And also there was also a correlation with precocity, because you said the insect arrives at a certain time, they eat pollen, and then they mate and put eggs on the pots, and then the larvae penetrate into the seeds. So the phenology of the plant, if you have a range of variation, also is not resistant, it's escape. So in that case, we could have an influence on the phenology. So having said that, yes, there is a correlation of seed color, but it was not strong, and it was possible to identify the tolerance into affection with nice quality.
Okay, a couple of questions from the chat. A quick one, is rust a big issue in fabas in Spain?
Yes.
And then I read also the second one. Okay, no, yeah, please answer.
I read, so this from Nicholas Larkan.
Yes.
I started working, I mean that when you say faba, mean faba bean, Vicia faba, right? Where we worked more in faba bean, we started working on rust in faba bean. It's an issue, but as let's say, it's taking, let's say, 20% of the yield. So farmers are not that much concerned, because if there is a big problem, they can buy fungicide, not very expensive. We have been working and we have developed a cultivar resistant to rust in faba bean. But make clear that the rust of faba bean is not the same rust that infects pea. So faba bean is Uromyces viciafabe that can indeed affect also pea, but only little. So can a lot under control condition, but seldom progress in the field. What we suffer in the field is from Uromyces piscis, that is a different species. I know from India that they might have different species. So when it is indeed Uromyces viciafabe, but the estial stage, what is infecting pea in India, but not in Europe, is, and I think in temperate areas, is Uromyces piscis. And then there is another question from Nicholas Trent.
Yes.
I have a question. I was wondering if you could give some detail of the nature of the resistance to Fusarium ostisporum race 1. No, I cannot give. I mean, you have there I gave the presentation. Please go to the presentation two publications where it is explained. Here I am focusing many different diseases, and I was focusing more on the identification of new sources of resistance and to use in breeding. Not at the level of the candidate gene, and it's not because I don't want to be cooperative. If you remember, I put the slide that was not clear because I said this is submitted paper, not yet published. So that is in that paper that is not yet published. So please wait a bit. And then I have another question.
Yes.
From Aphanomyces root rot.
I think the question is about how much introducing resistance will cost in terms of yield. The question is about a specific, specifically about Aphanomyces root rot, but I guess that would be in general if you can give estimates. It's really about how much does it cost in yield, how much is the expected reduction in yield.
As I said, Aphanomyces was not our expertise, and we just started to work on Aphanomyces because always when we was talking about RAS, Ascochyta, people were asking on Aphanomyces, so we started to work. I cannot give the details on the yield penalty or whatever because, as I said, we don't have Aphanomyces here in Spain. What I learned from people in Canada, in France, in Denmark, because I think that these people from Nordic seas here, that is from Danish German, Danish company, they are very much concerned on Aphanomyces, particularly because there is not resistance and the spores stay in the soil for different years, and then once you get the disease, you can go again, or you will need 2 long rotations. But I cannot say whether it will be 100% yield penalty or 50%. That is in the literature.
Yeah.
So, but the good thing is that we have identified resistance. I think bigger than the resistance reported and that it's being used. So there are nice work on QTL introduction of QTL resistance from to Aphanomyces in 2P done by people from INRA France, probably in Canada. I think we have identified a stronger resistance, but as I said, it's from a wild relative, so you will need more cycles of crossing to keep the resistance of selection, to keep the resistance and to get rid of the bad associated undesirable traits.
Okay. Other questions? People thanking you for the answers. If there's no more questions, well, it's now time to. So thank you again, Diego. It was a really interesting webinar of yours, and I think it's the right way, it's the right approach for disease resistance, as your results show. Okay, so to everybody, we'll see again in a month, and the webinar will go online between today and tomorrow on our channel. Okay, see you and thanks.